Hey everybody! Question for you. How amazing is this event? Brilliant, huh? And you know, out of the 8 billion people on the planet, these are the 1,000 I love the most. We're the best over here, right? And well, to finish this first amazing day, we got 2 fantastic talks and 1 panel, okay? So I'll start introducing the first speaker. We're gonna focus on the brain. The 2 talks will focus on the brain and how our most precious organ, our brain, can impact our longevity. So starting with Dr. Doug Ethell. He's the CEO and founder of Leucadia Therapeutics, a company that combines bench science, clinical studies, and machine learning to develop diagnostics and therapeutics for neurological disorders. After 20 years as an academic, he shifted to the private sector in 2017, 6 years ago, to focus on his company's technologies. In addition to more than 85 peer-reviewed academic papers and presentations, Dr. Ethell recently published the novel, Remembering Apples, A Race to Cure Alzheimer's Disease. So, Dr. Ethell, can you join us on stage? Thank you so much.
Thanks.
There you go.
Thanks, Dara.
See you soon. See you soon.
Hi, everyone. It's my first time here, and I'm having a great time. So let's get going. We work on a fundamental mechanism for brain health that removes insoluble metabolites, detritus, debris from the spaces in between the neurons, the interstitial spaces of the brain. Aging deteriorates this mechanism, which allows metabolites to accumulate, triggering inflammatory responses and a spectrum of pathologies that occur in Alzheimer's, Parkinson's, and frontotemporal dementia. Okay, today I'm going to talk about how aging of this mechanism triggers Alzheimer's disease pathology and how we can fix it with a tiny implantable device. So let's start at the beginning, around 1900, with a German woman, Auguste Dieter, who began acting strangely. She was confused, had trouble remembering things, and frequently got lost. These days, we'd call that mild cognitive impairment. She became worse, paranoid that someone was trying to kill her, so they took her to the Frankfurt Asylum, where she was seen by Dr. Alois Alzheimer. He diagnosed her with senile dementia, common in the elderly, but she was only 51, so it piqued his interest, and he followed her closely until she died in 1906. Now, Alzheimer, in addition to being a clinician, was a gifted neuropathologist. So when he examined her brain, he found, as you can see on the right side of the image, a wasting away or atrophy of the cerebral cortex due to the loss of billions of neurons. He also found these little Eiffel Tower-shaped structures called neurofibrillary tangles, as well as these waxy deposits in between the cells in the interstitial spaces of the brain. Now, he published this in a tiny article called Über eine eigenartige erkrankende Hirnrinde, which means on a peculiar, 3 years in Germany, that's no problem, on a peculiar disease of the cerebral cortex. And it wasn't until a year later when his boss, Emil Kraepelin, who published the big scientific text of the time, he referred to the unnamed syndrome and referenced it as Alzheimer's disease. And it's been called that ever since. Since that time, thousands of studies have been done on this, and we know a lot more about the disease. First of all, plaques, tangles, and neurodegeneration don't suddenly appear all over the brain. They start in one very specific area, highlighted here in orange. So this is the brain. If we cut it down the middle and open it up, so we're looking at the medial surface, right? At the bottom is the temporal lobe, and so that orange area is the medial temporal lobe, right? This area is important for sense of smell, making new memories. It doesn't store the memories, it helps make new memories, right? It tells us what to be afraid of and what's novel or what we've seen before, okay? So the pathology takes a few years to get going there. It's a bit like making a campfire. There's dried leaves and twigs, a little bit of smoke, and then poof, after a few years, it turns into a self-perpetuating pathology that spreads to the adjacent limbic areas, eventually consuming the entire cerebrum. It's a bit like a campfire that turns into a California wildfire. Now, the big question's always been, what's so special about this area that seeds the pathology in the first place? Well, it has to do with a fundamental mechanism that all tissues need to perform, and that's clearing their interstitial spaces in between the cells. So think of a stream in a forest. Leaves fall to the ground and they accumulate on the ground. But if they fall in the stream, they get carried away, right? Later in the summer, the stream starts to dry up, but the leaves keep coming. Eventually, a point is reached where more fall in than can be carried away. So if you look at the picture on the left, in the bottom, there's a rock in the stream, right? Imagine that's a cell in the body. It's in homeostasis. Things are being cleared away. And then you look at the cell in the middle of the image on the right, that's surrounded by a slew of metabolites that aren't supposed to be there, right? And it causes the cell to behave inappropriately. So the protein breakdown pathway, the ubiquitin proteasome pathway might stop working. So you get polyubiquitination of alpha-synuclein, which would aggregate into the Lewy bodies you find in Parkinson's and Lewy body dementia. Or it might impact the phosphatases that dephosphorylate intermediate filaments like tau, which would cause neurofibrillary tangles to form. So the environment that the cell in is very important. Okay, so the fluid in the body that clears the cells, it starts in the smallest blood vessels, or capillaries, that have tiny holes or gaps between the cells that are small enough that plasma can ooze out into the spaces of the tissue. There it's called interstitial fluid. Now it flows between the cells, picks up insoluble metabolites, pieces of dead cells, and any cancer cells that are metastasizing get carried in this system as well, right? So it goes to the open-ended lymphatic vessels that take it up, then it's called lymph, and it goes back to the bloodstream. Now, this system works very well, but if there's a problem with it, there are some proteins that accumulate that form amyloid folds, about 20 of them in the body, right? At high concentrations, they'll wrap around each other like carpet fibers, making fibrils and making things even worse, okay? That's called amyloidosis, and can happen in many tissues in the body, most tissues. But when it happens in the brain, we call it plaques. Okay? In the brain, there's also a little bit different system here because the tiny holes that are in the capillaries can't form in the brain. The blood-brain barrier protects the brain from blood-borne pathogens, so there are no holes, but the brain still needs to perform this function of clearing the interstitial spaces. And it does it by making a special fluid called cerebrospinal fluid, or CSF, in these, as you can see, these ventricles in the brain. So in the middle of the brain, there are these 4 chambers or ventricles, and they produce about half a liter or a pint of CSF per day, and it percolates out slowly. And this slow bulk flow clears the tissue and carries that stuff to the surface of the brain to a thin compartment called the subarachnoid spaces, okay? Now, the area where Alzheimer's disease begins would be the temporal lobe on the bottom. If we look at that from underneath, it looks like this. So on the bottom right is the temporal lobe. So the fluid comes up and it goes to the outside, which is now green arrows, right? It moves forward to this rudimentary funnel formed by the olfactory stria. Then it follows that down over the basal forebrain, which is another important area that's hit in Alzheimer's early on. And then the fluid goes up to the olfactory bulb. Olfactory bulb, well, that sits on a funny little bone, porous bone that's on top of the nasal cavity. So it makes the roof of the nasal cavity right between your eyes. So it's only about this big, right? And in there, the olfactory bulb sits there, and the cribriform plate is this bone, and it's got holes in it. Those holes allow the odor receptors in the nasal mucosa that pick up smells to send a signal up to the brain, to the olfactory bulb that receives those signals. Okay, so the fibers go through these little holes, and the CSF goes the other way. So let's take a look at this. We've looked at over 600 human cribriform plates, more than anybody ever. Okay, this is what it looks like from the brain side. So it's 2 little depressions, right, with holes in them, and there's a median eminence in the middle, okay, called the cristagalli, okay? And we looked at the porosity of those with age, and we found that it's pretty stable until you get to the 5th decade of life. And then it starts to decrease, right? You also lose a sense of smell starting at that age as well. So here's a heat map of the same idea. People on the left are 55 and younger. People on the right are 65 and over. And this is the probability that you'll see an aperture in any one spot in hundreds of subjects, from hundreds of subjects. So you can see the probability decreases as you go between these two age groups. Shown another way on the right are the gray bars. You can see the drop from 55 to 65. And then the people on the far right, those are people with Alzheimer's disease, right? They've got even less porosity. So this, you can read it all in a medRxiv preprint that's online. So the question is, how does the fluid get through there? Well, on the top here, this is a high-resolution micro-CT scan of a piece of cribriform plate. So we put 2,900 of those together to get the image on the bottom. Right? You can see the red or the blood vessels. On the bottom, you can see the holes in the apertures in the cribriform plate. And the gold or yellow-colored structures, those are the nerves that are coming up from the nasal mucosa to the olfactory bulbs, which would sit in there like two little sausages on top. Okay? So if you look at the image at the top, in the middle are two white bands going up and splitting. Those are the olfactory nerve fibers. They're going up to the olfactory fossa. But if you look at, on the right-hand side, the SAE, that points to dark areas. Those are fluid-filled channels that are carrying the CSF from the olfactory bulbs down into this system, draining the CSF, okay? And it's kind of a more complicated route for them because it's not straight away. So this on the right shows the conduits that are actually running front to back in the image on the bottom left. So all the apertures are connected together. And on the left-hand side of that image, there's a big cavity inside the crista galli. So if we put it all together in a negative image, what we've got is a structure like this. So the CSF is going into this structure, which forms a wonderful one-way valve where CSF is removed from the brain in a measured way, and it prevents pathogens in the nasal mucosa from getting into the brain. So it really protects us in two ways. Okay? So what we're saying is that these aging mechanisms we're looking at are damaging and deteriorating this cribrose watershed, and that's really what we're interested in. So that's a hypothesis and a lot of great associative data, but correlation does not imply causality. So what we did was test this hypothesis in an animal that never gets Alzheimer's disease, ferrets. Okay? Our team of human neurosurgeons went in there and made a little window in the snout, pulled tissue off the cribriform plate, and sealed those holes with dental cement. The ferrets were fine. They went back in the colony, played with their friends for months. And we checked them for spatial temporal memory, right? Getting lost, because there are cells in that area that I showed you, the orange area, that orient us in three-dimensional space. Most Alzheimer's cases emerge because grandma loses her way on the way home from the store, right? And the family spends hours trying to find her, right? Because those place cells, they get disoriented. So we check that in the ferrets by running them through tunnel mazes. Now, ferrets chase rabbits down tunnels. They kind of dug this study. So here's the two groups. The blue group is the control group between 2 and 6 months, and on the y-axis, that's how long it took them to solve the maze. So you can see it's going downward to the right. They're kind of figuring out some shortcuts of how to solve the maze. They've got it figured out. Now, the occluded ferrets, that's the orange one, and the trend line is the dashed line. You can see it's taking, they're not figuring it out. It's taking longer and longer. They've got a progressive memory deficit. Now, a really startling thing we found was when we pulled out the brains. Okay, at the top of this image is that human thing I showed you. Okay, not sure if you can see it here, but in the two ferret images, right, these are the brains from underneath. The green outline, that's the medial temporal lobe of the ferrets. On the right is a control one, which is big, and you can see the little grooves in there. But on the left-hand side, it's smaller and atrophied. Right? We also got atrophy of the cribriform plate or the olfactory bulbs and the fiber bundle that connects them. It's a 40% loss, right? And this is not attacking those parts of the brain. It's just blocking CSF way up here, and we're getting this effect down here, right? Because of the CSF clearance. So we think that was pretty compelling evidence that we were right. Now, how do we translate into something that helps people? So what we did was a Project Cribros. We looked at 560 people between 20 and 90 years of age. Okay? They sat in a dental scanner, which takes about 20 seconds. It's low dose, goes around the head, and then we can see the cribriform plate. Now here's an example of this aging in progress. All right? 3 women, all in the same family. In the middle, orange highlight, that's a mother. She's in her mid-50s. On the left-hand side is a daughter in her mid-20s. And on the right-hand side is a grandmother in her mid-70s. Now look at the 26-year-old on the left. Do you see the apertures on the left? They're open and clean and nice. Look at the ones in the middle, and they're a little smaller, occluded. And the ones on the right, many of them are completely occluded. So we've got this slow occlusion. It's closing it off. Okay? We also check them for memory and smell. Okay? We did that with we sort of improved the standard, which would be a piece of paper and a pencil, you fill it out. We turned it into a video game. So it's a touch screen, takes about 15 minutes. It's a series of little games. Here's an example of one. Okay? It's a recognition, object recognition. So you'll see silhouettes of 3 animals, right? And this plays much faster than it would ordinarily do. And then which ones did you see? Well, those ones, right? So you play games like that for about 15 minutes, including mazes, so we're measuring spatial temporal memory. And what we got is, for our entire population, is these are decades, okay? So the one right down the middle, the ones in the center, those are people in their 20s, right? They make very few errors in these things. And the outer one, that's people in their 90s. They make proportionately more errors. And the other bands inside, those are the decades, right? 30s, 40s, 50s, and so on. Okay, so how did these 3 women fit into that plot? Well, here they are. The youngest one, green, you can see she's doing okay. Speed, she's not so fast. Whether that's a TikTok thing, we're not sure. And then the orange one is her mother, right? She's also not so quick. Maybe it's Facebook. And then on the bottom left, that's spatial temporal memory. That's solving the mazes. She's having problems with that. Now, the grandmother is the blue one. Not sure if you can see, there's a little dashed line in there. That's what we would expect for someone of her age, right? But she performed more poorly in the smell, so she's lost her sense of smell, and also spatial temporal memory. So she's the ideal candidate for an intervention that restores the CSF flow. Actually, her daughter probably is too, for preventative. And so how we would solve that is we've developed a tiny device that goes into the cribriform plate. It's an inlet, allows fluid to go in, follow a little tube to a tiny pressure regulator that goes in the nasal mucosum. So it's not dangling in the nose, and it allows a measured egress of the CSF. Really, it's just restoring the system that's been working up until that point. Okay? It will help clear the CSF in the blackened areas here, which is where Alzheimer's strikes early on. Now, these areas overlap also with another syndrome that happens in younger people from 46 to 65. It's frontotemporal dementia. You may have heard Bruce Willis has this. Same areas, more frontal lobe than temporal lobe. So we think this might also work for them. And another syndrome, maybe you've heard of COVID? Sure. Okay? Remember, it starts with infection and inflammation of the olfactory system, loss of a sense of smell, right? And in people who have long COVID with cognitive brain fog, right, they might have had scarring of the Cribros watershed that prevents the regular egress of the CSF. So it'll be very interesting for us. Hopefully we'll get in clinical trials before then, because if we're right, then about 5 years from now, those people with a long COVID are going to start turning into Alzheimer's patients. And it's going to be a real mess. Okay? So these are our plan for our clinical trials. You know, first we'll do Alzheimer's, long COVID, FTD, and then brain aging. This little device is so small and unobtrusive, we could put it into the parietal lobe, occipital lobe, and drain CSF, increase CSF drainage in other parts of the brain as well. But it's a lot easier once we have an approved device. So in the spirit of RAADfest, I have a longevity hack for you, right? So let's say you don't quite have Alzheimer's yet. You might be able to improve your CSF flow. So this is how the brain actually moves with the heartbeat, right? So it pulsates, right? and it helps propel the CSF outward, right? Out to the subarachnoid spaces, highlighted here in blue. Now this is a cartoon. It's much thinner in reality than it is in the cartoon. Some areas are little cisterns or pockets of fluid, and it doesn't mix very well when you're sitting in a chair staring at a computer or watching TV, right? In a sedentary lifestyle, right? Maybe when you lie down, you get better drainage because you roll left and right. You get a little angular momentum mixing, right? So areas that have a higher proportion, higher density of metabolites will mix with the others and you'll get better clearance. But remember that the outside goes all the way down the spinal cord. So the subarachnoid space goes all the way down to the lumbar cistern, which is where you get a spinal tap from. So what you've really got, everybody, we've got a 2.5 or 3 foot water column of CSF. There's another way to mix that up. If you take yoga and you do a downward dog, basically it pushes the CSF down and it will mix up the CSF going around the brain, right? Now, if yoga is not your thing, you might do what I do, which is incline sit-ups in the morning. But instead of going up right away, I sit there for a minute, turn the head left, turn the head right, lift it up, push it way back, and I'm good to go for the day, right? If that's not your thing, you could always just get an inversion table too, okay? Now, I don't have any evidence to show and prove that this will improve your brain health, but I know a lot about this stuff, and I do it every day. So yeah, that's that. And then I'd like to just acknowledge everybody who's helped us along the way. Our clinical trial will be at Eisenhower in Palm Springs, which has quite a sizable retirement community. And our first funding came from Methuselah. Happy to take questions at the end in the summary. But thank you.
There you go. Q&A. Thank you so much.
All right.
Thank you so much. That was brilliant. Oh, you need it? Perfect.